there. So the idea in the next 10, 15 minutes, I would like to give you an idea what are we doing in Viralgen and why we are doing this kind of thing. So basically, in a very basic idea of the gene therapy is just adding new genes to replace or replace mutated gene. No, this is the basic definition for gene therapy. No, and we can do it in several ways. We can do it yes with the naked DNA. This is the most simple one, and of course, it's done in some kind of therapies. But the problem is that the body is designed to somehow destroy any naked DNA that we can introduce. Of course, the body doesn't distinguish between bad and good DNA and is destroying the whole DNA. So that is the reason that some companies has decided to somehow encapsulate this DNA into nanoparticles. So when we introduce this into the body cannot see properly the DNA and this get into the cells. And the problem with this nanoparticle right now is sometimes they don't have the access to the nucleus, so they can translate, or sometimes they're inside of the cell, they get destroyed as well. No? And the last thing that we can do as well is just to use virus as a tool to introduce DNA into the body, which has also a lot of problem because our body is designed to destroy all the virus or bacteria that we are getting from outside. No, but the thing is that the Viralgen is concentrating in how we can manufacturing this kind of virus for gene therapy. No, and basically this virus. If we look at all the virus in nature, well, not all of them, but some of them, we have virus that they produce AIDS, like HIV. We have virus like adenovirus that produce some kind of cold, influenza for flu. We know already COVID now. So we have learned so much about this virus in the last 20, 30 years that now we are using some of them to introduce, like I said, DNA or genes into the body, no? And when are we using one or the other? Was basically when it's not always the same, but most of the people that they are trying to work with blood system, the blood, they're usually they're using HIV. So most of the people working in cancer diseases, they are using mainly adenovirus. And now we are concentrating basically in another very small virus, which is adeno-associated virus, that we have decided that the probably is one of the best. No, everybody is infected with this virus. Doesn't produce any disease at all. So we get infected, but we don't have any disease. So the virus, we don't even realize that we have the virus inside. So that is the reason that the probably in terms of security is quite interesting, this virus. No. So how we are using this virus? So basically, I guess you already talked about that. This is an example, for example, of if a child that has a mutation, a mutated gene, no? And when you have a mutated gene, you basically, in a normal situation, normal gene gives you a normal protein, but when this mutated gene, you can have a normal protein, basically that has no function, or really low function, or basically you have no expression of the protein. So you don't have the protein, so or very low expression of the protein. So it's when you basically have the disease, no? So where are we doing when we are using virus for gene therapy? So basically what we are doing is to use this virus to introduce the normal gene back into the patient, no? And we are usually overexpressing the normal gene through to the mutated gene, no? This is most of the therapies that we are doing at the moment are based in this idea, no? However, we can see now that more and more companies that they are using another strategy, which is edit the genome. So now we are not to overexpress the gene, but basically what we are repairing the gene inside of the chromosome, inside of the DNA. So obviously, there is no doubt that this is going to be the future, because as soon as you have a specific in the place that you have the mutation, you can repair everything. So the gene is going to be regulated through the whole system and as a normal gene. So this is going to be the future, but we still have to learn quite a lot before we can start doing this kind of thing. No? I would like to present you, this is the virus that we are working. It's a very, 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 very small virus, basically 23 nanometer. It's so small that it cannot have the whole genome inside. So every single time that he has to infect a cell needs another virus to have the life cycle. And that is what we call adeno-associated virus, because every single time that is infected the cell needs another virus to infect the cell. No? So we have, inside of this virus, we have the DNA, and we have specifically the DNA that corrects the mutation, like I say. No? And how we are doing, what is the theory behind that? I mean, we don't need to know a lot of biology about that, but basically is we have a virus that in the normal life, we have a virus. In this case, like I said, needs another virus. So this co-infection give into the cell, give the possibility to virus to replicate, to form more virus, and to go out. No. So like I said, we have learned so much in the last 20, 30 years about these viruses that now we are taking the pieces of DNA of all these viruses, and we are putting in a different position, in a different formats. And now we introduce all the DNAs into the same cell. So basically, now the virus thing that is replicated in cell, but basically what it's doing is replicating whatever we want to have inside of the virus. So this replication replicate many, many times and then encapsulated, and all the virus that we are producing, that the virus thing that has its own genome, basically what is having is the gene of interest that we want to repair, basically. And this is basically how we are producing, in theory, all the virus in the lab. But basically, this is the theory, how we are doing this in the lab. So, and before we get into that, we need to understand what are our challenge. No? And one of the challenges that I put two examples of eyes disease or muscle disease, which are the best and the worst case in terms of gene therapy. And this is the gene therapy is a dose-dependent therapy, like most of the therapy that we have. And basically, the doses that we are using is a 1/10 to the 12 virus per eyes when we have to repair a gene inside of the eyes. But when we go to the worst case, is like a muscle, we have to introduce 1/10 to the 14 virus per kilo of patient. No? If we have a normal patient of 70 kilos, so we have to regenerate easily, we have the mathematics, we have to introduce 7 to 10 to the 15 virus per person. No? When we go to the lab and see, well, what is in a normal life, we are using a cell line that I'm going to introduce right now. This cell is able to produce around 1/10 to the 11 virus per ml. So if we extrapolate this into the needs that we have, basically for an eyes, we will need 10 mLs of the culture that we are producing. However, when we have a muscle disease, for one single patient, we will need to produce around 70 liters. No? And that is the main challenge that we have when we are producing the virus. No? When we go to the lab, this is I introduced you, this is the cell line that we are using, just some cell. We are putting the DNA here, and then the cell produces the virus for us. No? And when we have a look, this is the typical plate that we are using in the lab, no more than 5 ml per plate. And you can imagine if we have to escalate up to 70 liters per patient, it will be a nightmare. No? So some people has decided to use this kind of cell stack that they give you no more than 5 liters. So when we start thinking about how we are going to treat several people, several patients, now you have to start thinking in this kind of culture. No? We have decided to do another strategy, and the strategy came from this guy, Josh Grieger, that in 2010, he took these cells and he stressed it in a way that the cell now, rather than to grow adherent to the plastic, now they are growing in suspension. This is very important because now, rather than to work in 2 dimensions, now we are working in 3 dimensions. So this is a completely different. He also did a lot of things. He removed all the serum that we are using to grow these cells. This is important in terms of securities, in terms of some kind of bacteria or prions that we can introduce into our system. And of course, has a high transfection capacity. And this is the cell line that we are using to produce the virus. No? This is the typical work in the lab that we are doing, no? The people working to produce the virus. This is a typical bioreactor. For example, this is a 250-liter bioreactor. So here you can see the cell, as soon as we are lysis the cell. So now we just lysis the cell, and inside of here we have million of viruses. And of course, after that, we cannot use that. We need to purify all these media and to reduce the volume, of course, if we want to inject it into the human. But this, I just wanted to give you an idea how we are working in a lab, no? The next problem is that we are growing this cell, we have to purify, and then we have to fill the drug product inside of this vials, no? However, one of the big problems with all these things is that in every single step, we have to be sure that we are doing in a proper way. So we have to introduce all this quality control step by step. And this is the reason that become really, really complex. It's not just the difficulty of growing cell in a scale that is really, really big, but also all the quality has to be qualified or validated. So we are completely sure that when we are introducing this virus inside of a patient, is always in the same way and is secure in somehow, even though we are at the really beginning of the gene therapy, no? And we don't understand most of the things, but at least all the qualities that we are having, we need to have to do it in a proper way, no? And this is just to introduce how we are working, no? In a normal lab, we can work like even you are doing almost every day the same thing, but not in kind of the same way, no? Even though you can have the same flavor. This is a typical, when you go to the regulator and you ask, oh, I want to do the virus, they are asking you to do always in the same way. And this is what we call GMP thing. And in fact, we need to have a GMP accreditation from the regulatory agencies to be able to produce virus to inject in human, no? And this is regulated by, in every country is different, but the main one are FDA in the States and EMA in Europe, no? So, and the last 2 minutes, I would like just to talk about a really difficult idea, which is the amount of money that right now the gene therapies need to start with all the therapies. And basically, everybody knows that these gene therapies are really expensive, no? And what I would like to say is that we are at the beginning of these therapies, and that is one of the main reasons why these therapies are so expensive. I'm going to give you 3 examples why. What is the reason for that? No, the first thing is the complexity of the drug. No, so basically, long time ago. We were starting working with a small molecule. Now it's like an aspirin. That was that is quite easy, even if it's complex. It's quite easy to synthesize and to work with. Now a few years later, we started with some small biological as well. Now the complexity, as you can see, is becoming a little bit more difficult. And then in the last 20 years, we have been working with another molecule, which is antibodies. And as you can see, the complexity now is much more than the other. No? If we are now talking about cell therapy or gene therapy, we increase like several logs the complexity of the drug that we are manufacturing and we are working with, no? Because now we are working with a live organism. That is, there are no more just chemical compounds, no? And that is one of the reasons why in the recent analysis, the cost of gene therapy can be between 1 to 2 million per dosage, no? This is something that like I said, I mean, we don't have to be really worried about that. It's a question of time that we are going to decrease that price. Just to give you an example, 20 years ago, we were able to produce a few micrograms per liter of culture of antibodies. Now we are producing several grams per liter of antibodies. So this is something that is going to come, and it's a question of time. But like I said, we're at the really beginning. The second thing is that the manufacture is really expensive. I mean, we are working with a raw material, which is the DNA. It's the DNA that we are introducing inside of the cell, and this is really expensive. I can tell you that a gram of DNA right now is more expensive than gold, no? Just to give you an idea, no? And then the second step is the scale up of all the manufacturing, no? You think, well, it will be really easy to go from 50 to 250 to 500 to 2,000 liters. And I can tell you that this is not linear. We have to do a lot of experiment and really specific experiment to be able to be completely sure that we are producing at a small scale into the bigger scale that we can have, that we have right now 2,000 liters. So every single time that we run 2,000 liters is almost more than 2 million euros, and we are facing many problems. So not all of them are available to the patient, no? And the last one, which is really important, is that we are facing a completely different new paradigm. Which means that we are working as a single dosage. So this is, for a pharma company, this is not ideal, no? Because the pharma company, what really wants to have is just to sell as many drugs as they can. And now we are facing something that with one single dosage, we are able to treat the patient, no? And now we are working mainly in rare diseases, which means that there are very few patients. So basically, the investment in manufacturing is massive to have a single dosage for a few patients. And that is one of the last reasons that I think is why these therapies are so expensive. But like I say, it's a question of time that is going to be a reduction in that. And I think from this one, I really say thank you to everybody and thank you for the invitation.